Thinking Aloud. Conversations on the leading edge of knowledge and discovery with psychologist Jeffrey Mishlove.
Hello and welcome. I'm Jeffrey Mishlove. Today we are going to explore psychic healing research. And with me is my dear old friend, Dr. Elizabeth Rauscher, a pioneer physicist, author of several books, including Orbiting the Moons of Pluto, Complex Solutions to the Einstein, Maxwell, Schrodinger, and Dirac Equations, and also The Holographic Anthropic Multiverse. Dr. Rauscher was featured in a book by MIT professor David Kaiser called How the Hippies Saved Physics, Science, Counterculture, and the Quantum Revival. Welcome, Elizabeth.
Thank you very much, Jeff. It's wonderful to be back with you and to explore where we're going now with what we did earlier. And it's just great to be here.
It's a pleasure to be with you. You and I go back a long time. I remember the time I first met you when you walked into the Institute for the Study of Consciousness where I was living in 1973 in Berkeley.
Oh, yes.
And since then, you've done an enormous amount of research in many different disciplines. You've published, I should say, about 250 scientific papers, or should I say more than 250?
More than, because I have to keep track of them all.
Yeah. And what we're going to look at now is some of the classic studies that you have done with Dr. Beverly Rubik in the area of psychic healing, working with a well-known healer of that era, Olga Worrell.
Yes, I'm very interested. What I did is, in my endeavors in psychic research, I'm an open-minded skeptic. Now, what I mean by that is I'll consider all possibilities, but I apply this to conventional physics as well, or conventional science, or conventional ideas, because you need to be open-minded to all kinds of possibilities, but skeptical and not just accept everything. So with that in mind, Dr. Beverly Rubik and I designed an experiment with Dr. Olga Worrell to...
She's not a medical doctor, we should say.
No, she had an honorary degree.
Of theology, as I recall.
I think it was a theology,
but
yes.
She's a very well-known healer.
Very well-known Russian aristocrat. And so what it was that I had the opportunity through John F. Kennedy University, then in Orinda, California, to work with Beverly, to work with Olga. So immediately I thought, well, all the mechanical PK kind of devices that she might affect, she's not into that. She's into biology. She's into healing people. Yeah. So Beverly Rubik was part of my fundamental physics group that's in David Kaiser's book. So I asked her whether she would participate with me, and we had a little grant. And so then we designed this experiment to look at the motility or movement of bacteria and growth rate. And so she chose Salmonella typhimurium, which is a mild pathogen, and Escherichia coli, because when you grow the bugs, the Escherichia coli stinks after a while. So the Salmonella may give you gangrene or something, but it won't stink. And so I had not met Olga at that time, and she came out.
She was based in Baltimore, as I recall.
Yeah, she and her husband, her late, she's deceased now. She and her husband had a wonderful place in Baltimore, which I later visited.
A healing church.
A healing church. And there were all kinds of stories. Like she would have people come up and she'd do a laying on the hands healing. And she'd do it for about 2 to 4 minutes. And so she had people come up. And she had this one man that had a tumor on his face, a really big tumor. And the statement was, even by MDs, that they just saw the tumor disappear, sink in, disappear, and the cancer was cured. And so there were all these kind of stories. Well,
And she was very open to working with scientists.
She understood the scientific method. And I'll tell you more about that because it's fun. But what I felt was, instead of working with humans, we were going to do, you introduce a controlled wound, and it's just too complicated. To look at a simple bacteria system that's quite interesting, and what salmonella and other bacteria do is it's called chemotaxis. They move towards food and away from shit. They they basically are environmentally conscious. They're environmentally conscious. They go towards the good stuff and away from the bad.
Yeah.
Anyway, so she came out, and we're set up at UC Berkeley Life Science Building to do the experiments. Well, of course, she comes in with her pillbox hat and her mink coat, and we're trying to smuggle her in. So we're trying to throw a lab coat over her. And she comes into the lab, and what we have is a room that's 37 degrees centigrade, which is the optimum growth for the bacteria, and then a control system down the hall that she won't interact with. And she said, well, I hope you got controls, dearie, because that's the way she talked. And she understood to not affect the controls, to infect what she was treating. So we had multiple test tubes sealed against any physical contact. I mean, we had all the protocol down, and Beverly was terrific. I mean, this was her landmark experiment that she had done for her PhD at UC Berkeley in biophysics. And so it's the first psychic experiment she had done, but she caught on right away quick, And it's a lot of things. It's about making a positive environment. It's true of any experiment.
Well, there's an irony in this particular experiment, in that you were asking, if I understand it correctly, Olga Worrell essentially to send healing energy to these bacteria, which are, as you say, mild pathogens.
I asked her about that, and she said, we showed her in a dark field microscope, and they look like little light dots moving around. And she said, oh, they're cute little critters. And so I explained to her it's a mild pathogen. So she says, I heal the highest form of life I'm near. And so they're the highest form of life I'm dealing with, so I will only heal, which is interesting, because I'll tell you about working with another healer on the same experiment, a similar type experiment. So what we introduced in motility, which is the movement, and you can see a movement like little dots.
The more they move, then they more alive and happy they are. The more
happy they are. Basically, it's about being happy. And really the question is there an optimal state of health for any organism? Is there a state in which we're normalized, we're reasonably in good health, our ATP metabolism and everything's working right? And I think there is. I mean, this is a question that Beverly and I asked. So in the motility experiments, you would introduce chloramphenicol, we use tetracycline, chloramphenicol, and phenol as antibiotics. So we made them ill.
Yes.
So we show her the well ones, and then we add phenol. Now, they will remain motile for about 12 minutes until the slide in the microscope dries out. And so they're little light dots that move around, and they get fewer and fewer as time goes on.
And you have a precise, I guess it's a spectrographic analysis to determine exactly how active they are. A Zener
light source and a calibration, so it counts the number of moving dots. And it's like looking at particle tracks, really very similar. How long they are, how many little dots there are, and so forth. So so then we add the antibiotic, and what happens is they die in about 1 or 2 minutes. So there's no motility in the control. In the treated, what she did is cup her hands near the slide, but she did not touch it.
Yes.
They remained modal for 15 minutes with a good count number. Amazing. You know, it just was, and she says, I hope you keep these slides forever. Well, the thing is, of course, they're useless after 15 minutes because it dries out. So Beverly put them aside, but made her think that we were going to keep them. And
but of course, you kept the record of what occurred.
We kept the record, so we had all that recorded. And it's very interesting because motility is fast-moving. Growth is slower, so you're measuring like the rate of enhanced health as well as the enhancement of the health. So you do different doses of the antibiotics. So in the growth studies, we had sealed test tubes, 3 in the group. They were all coated with the controls coated and the treated coated, and it was really tricky because Beverly and I had a grad student code them, so we wouldn't know when we analyzed the data later whether it was a treated or control.
In other words, what we call a double-blind experiment.
A double-blind experiment. So motility, in the growth studies, you do about 24, you can do up to 48 hours of growth, which means every hour you have to go to the lab and measure the growth and a spectrophotometer. Now, the first one we used was a nice standard Beckman, which I've used before. Then one time we did 4 major studies with replication in all studies, which is very important. And all the controls were like the background of what's known for that bacteria with no intervention. In the treated sample, she would hold her hands there for 2 minutes. For each batch of test tubes, so we had multiple test tubes, so we had redundancy. It took me months to analyze the data, doing little graphs and stuff. And she was very, very cooperative, very patient, and very good at understanding the scientific protocol. So the first one was a Beckman spectrophotometer, and what you do is you take a sample from the test tube at each hour, and then you put it in the Beckman spectrophotometer, which measures the opacity. So the more bacteria, the more opaque it is to the light coming through. So we measured that, and then of course what happens is, if they're well, they grow in what's called the J curve. They start out slow and then they grow really fast exponentially and then top out when they use up their food source. So there's a carbon source in there for them to eat. And of course then, because it's a closed container, there's the excrement, which means that the waste is also influencing, and that's why they top out after about 48 hours. And it's a kind of a opalescent fluid, so it kind of looks nice in the test tubes. So the first test tubes we used were glass. We even used plastic, didn't seem to make any difference. And then we did a dose response. We added like 10 milligrams, milliliters of chloramphenicol, 20 and 40 and different amounts. So of course the more antibiotic, the more sick they became and the harder to heal. So that showed up as correct. And it also matched over the 4 major studies. So
now let me just backtrack a little, because I understand that somebody might say, since she held her hands near the test tube,
I got an answer for that.
There was a physical effect, heat or electromagnetic energy.
She had a hidden magnet in her tooth and stuff like that. Okay, what we did is take grad students that were blind to the meaning of the experiment and ran the whole experiment with grad students cupping their hands. We figured they probably weren't healers.
You never know.
But you never know. But we had them cup their hands and then read the whole experiment with someone naive to the purpose of the experiment to not introduce a healing effect, and they were just exactly like the controls. And there were some interesting predictions, like she said, there'll be extra growth in test tube number 20, dearie. Because that's the way she talked. We took the pill box hat off, a few things like that. But anyway, the first one was done at Life Science, and then I did a next one. We did it at the Lawrence Berkeley Laboratory, and we had a spectrophotometer that was worth about $500,000. And the whole side of a room would be covered with this instrumentation. And all it came down to is the little cuvette holders weren't as good as in the little Beckman, which is about like that. But so the arrow bars were a little bit bigger, but every replication was very well replicated with amazing effects.
And if I recall correctly, this experiment, you ran it over a few years.
4 years. We did 4 major studies over 4 years.
Like a year apart each?
Yes, partly because it was raising the funds.
Yeah.
And the Holmes Foundation of Los Angeles was one of our funders, which I give them credit for. A very nice group.
Related to the United Church of Religious Science.
Yes.
That's what it was known as then. I think they're now called the Centers for Spiritual Fellowship, or
I think so. Yes,
something along
but
those lines.
They were doctors and other professionals, so it was a very diverse group. You know, and then of course we reported our findings. But we did 2 more studies. One I had arranged to part of UC Berkeley at the Richmond Field Station. And then this is interesting because, I mean, Olga was great. The only thing we had a problem with is she had to fly first class, which used up about half the grant money. Yeah. But I analyzed the data, and Beverly did a wonderful job. It was a really good experiment.
Well, our viewers should know that this experiment, which is now a couple of decades old, it still stands as a classic of research in the field of psychic healing.
It's so controlled, so, I mean, I think that my design and then working with Beverly was so important. Now, other people tried to replicate it. Now, here's what happened. Of course, they had different healers.
I mean, Olga Worrell was really quite an extraordinary
classic person. You know, she was so cooperative. She was so knowledgeable. She was a really fantastic healer and a very lovely person.
And other researchers have worked with her and also reported quite astounding effects, as I recall.
Very much so. She did some work with like a cloud chamber and made it look like more particles were present. And then there was several replications. Now what happened
I think she worked with Wilbur Franklin, a physicist at Kent State University.
Yes, and he had done some very interesting PK metallurgical work with electron microscope work, similar to what we had done. And
well, we could talk for a long time about some of this other research.
What happened with some of the replications.
Yes.
They didn't instruct the healer that there were controls, so the controls were anomalously affected. So the statistical difference wasn't as great, but the controls didn't fit the baseline. So they were all healed, because it was like the target is to heal this, but anything like it looks similar to the healer, so psychically he could go down the hall 50 feet and heal the controls as well as the treated.
Well, you know, I know one parapsychologist has criticized a lot of healing research, saying just because you have a difference between your experimental group and your control group, how do you know that it was the experimental group that was healed and not the control group that was harmed?
Well, the control group matched all the previous many years
Beverly,
of study.
in this case, had been doing these studies for years and had a
good baseline. And also, her research advisor had done this work for 15-20 years. So actually, it had like millions of example baselines. So when the controls met the criterion of the baseline, I don't think there was any problem.
So, but you're saying in the replications?
In the replications, they didn't distinguish, but you see,
even
Olga
the control group
knew
didn't match the baseline, in other words.
That's the point.
Yeah.
And so what Olga said was, you know, you got controls theory. So she understood that 50 feet down the hall was an equivalent lab with the controls to not affect them. So that was a choice, like how far does the field go? What is your intentionality? How is it focused? How do you develop from the spiritual healing or psychic healing she's doing into the lab? So there was a lot of wonderful factors that we had to deal with that really made it work so well, and the people involved and our assistants were so wonderful and cooperative.
Now, there are many different views about psychic healing. Of course, some people think it's all suggestion. Other people might go so far as to say it's all fraud. Some people say it's about prayer. They're praying to a higher power. Other people say it's a psychokinetic effect, that mind over matter. The healer is directing energy according to their conscious intention, which seems to be the way Olga Worrell was working.
I actually explored that by talking to healers. Now, what I have found, the best healers say they bring the energy from a higher state. Because if you're just using psychokinesis, you may be using up your own energy. And often healers get ill. In fact, there's rituals like with the indigenous people and the medicine men, because they have to rid themselves of the disease which they take on to get rid of it from the person they're trying to heal. So all those factors come into play, and it's important to distinguish. But what I think is, on the idea of, the controls is how we presumably dealt with fraud. And the bacteria are not something Olga Worrell had no idea what system she was going to work with, so I don't think she brought any salmonella with her. And the reason I thought about salmonella is how suggestible are they? Are you going to be able to tell the salmonella, now you grow better because I'm telling you this? So they're not suggestible like people, and that's why I went with that, because there are other experiments where you can introduce a small wound, cover it with a plate, look at the rate of healing, but that depends on the different subjects' state of health. And we're doing 10 to the 8 bacteria in a test tube, and it would be a really huge project to just run 100 human subjects. So we answered that, but just in case, the night before, I asked the salmonella for the permission to do the experiment. So they'll say, oh, well, that gets into prayer, but I'm not sure that really influenced it.
Well, these days in parapsychology, I think it's widely understood that any experiment is a whole system. We don't know whether the experimenters or other observers might be exerting a psychic influence on the experiment.
Exactly. Now, let me give you some examples. When I'm mostly, my background's in theoretical physics, but I've done a lot of experiments. I had a whole chem lab. I built telescopes and cloud chambers, and I started building rockets, but then I went to college and didn't have time to get into that. But I've worked with a number of groups, both as a theoretical collaboration and experimental. And my idea with working with the accelerator at Lawrence Berkeley National Laboratory, the Bevatron, which no longer exists, is why not PK a particle? So you need to choose a particle. So I was going to do a Lee-Wicks vacuum state, see if I could get the grad students to PK it, and then we get a bigger grant. Well, it was partially a joke. But I did do some experiments with influencing the rate of radioactive decay. And it's like, I think every experiment has a psychic component. Now, of course, electronics, everything is hardened against psychic influence. It's linear, so the more nonlinear system is, like a living system, the more subject it is to influence by external mind effects. And so what I did is I did a 6-hour experiment, and I'd have them concentrate for 15 minutes, and I have them agree to slow down the rate of radioactive decay. So they all agreed to that. So the effect would be similar.
Yes.
Then I ran them around the hall so they wouldn't think about it in between, and then do another 15-minute session. So I got about a 4% effect. But there is a literature where there is deviation from the rate of radioactive decay, and it all bridges between about 2 to 5%. It's very interesting.
Yeah.
With different ways of doing it.
There's now quite a large literature on, on that sort of phenomenon. It's typically called micropsychokinesis.
Right. And I would say in the groups that worked at the Bevatron, there were some groups that just clicked. They really cooperated, and they worked well together, and others that didn't. And the difference in all factors, like the background noise and so forth, was majorly affected. I mean, so I'm not sure that you can't do an experiment and completely exclude psi. Because I asked the students and the professors, I said, what's your purpose in doing this experiment, the Bevatron experiments? And they said, I have no purpose. And I said, no. I don't believe that. I mean, you know, like the grad students, you want a PhD. Whether you want to prove it to your girlfriend you're smart, or your parents, or whatever, or get a job, or you love physics, there's always a motive. And that's very critical to look at in any of these experiments.
The human element.
The human element and all the humans involved in their intentions.
Yeah. Well, we have so much to learn about this, but you, Elizabeth Rauscher, have been a very important pioneer in the field. Thank you so much for sharing this half hour with me, and I look forward to future conversations with you.
Thank you very much, Jeffrey. I really appreciate it. I always enjoy talking to you.
And thank you for being with us.